The topic of Lecture 15b is protein interactions with substrates. Please notice that all figures and tables of this lecture are from Chapter 3 in the course textbook. The interactions of proteins with substrates, both natural and synthetic, have been investigated extensively by scientists, researchers in the fields of chemistry, biochemistry, chemical engineering, and, of course, biomedical engineers who are interested in implantable biomaterials. Pertinent research is going on as we speak, and it has addressed two aspects. First, the interactions of proteins with substrates involves studies at the laboratory bench, therefore experimental. In addition, however, many of our colleagues have explored those interactions from the computational mathematical modeling point of view. We do have some faculty in our UTSA Biomedical Engineering Department who are studying these interactions of proteins. Lecture 15b is a short introduction to protein interactions with substrates. Let us first consider the interaction of proteins with substrates from what I call the protein perspective. For this purpose, I will summarize the important results which have been brought to our attention as a result of extensive research, experimental research in this particular field. The most successful results pertinent to protein interactions with substrates were obtained when researchers investigated first very dilute protein concentrations, and number two, solutions of only one type of protein. We'll start with pertinent definitions. First and foremost, adsorption is a term used to describe the process during which proteins or other similar chemical compounds quote-unquote stick to surfaces of substrate. They could be either natural or synthetic substrates. Notice that I use the colloquial term stick and I put it in parentheses because I wanted to describe the concept, but from now on we are going to use in this course the term adsorption. Please keep in mind that we use the term adsorption to describe what is happening when proteins interact with the surfaces of substrate materials. In contrast, remember when we were discussing, let's say, the platelet interactions with substrates, we call that process adhesion. Therefore, there are two different terms. Adhesion is used for the interaction of cells with substrates. Adsorption is the term which is used to describe how the molecules of proteins interact with the surfaces of substrates. Several properties of proteins are known to affect their subsequent interaction with material surfaces. Some of those properties are given in Table 3.1 from Chapter 3 of the textbook. First and foremost is the size of the protein, and as it makes sense, a larger molecule will have more sides by which it can contact the surfaces of materials. This concept is illustrated in the cartoon of figure 3.2, which shows a big molecule of protein which has many more chances to interact with the surface on the top of these three levels compared to the lower level, which illustrates in a cartoon fashion as much smaller protein. That particular molecule will have much fewer contacts by which it can interact with the surface of the substrate material. Another aspect which has been recognized as an important parameter that affects the interaction of proteins with substrates is charge. Remember when we talked about the amino acids, the building blocks of the proteins, we mentioned that some of the amino acids are neutral as far as charge is concerned, but some of them are positive or negatively charged. As a result, the protein, which is now composed by many such amino acids, may have either positive or negative charge or neutral charge as well. Because of that particular aspect, the subsequent interactions of this type of proteins with the surface of material is affected. This aspect is illustrated in the cartoon of figure 3.5, and you notice the various little dots close to the surface of each one of those so-called protein illustrations can be either a negative or a positive charge. As a result of that, the way a particular protein molecule interacts with the surface, the orientation of that molecule is going to be totally different. Last but not least, and we're going to discuss this again later on in this lecture, the surface of the biomaterial may have its own charge or its own domains of different charges, and therefore that will be another aspect by which the two, namely the protein molecules and the biomaterial surface, will interact. Proteins are known as big, bulky molecules. In solution, however, they acquire a much smaller size, which becomes pretty much almost spherical. When they come into contact with the surface, though, as this particular figure 3.7 shows, the time-dependent sequence is that of spreading, namely, the molecule of protein, as time progresses, makes more and more contacts with the substrate surface. So you start with, on the top level, with a molecule which is very much rounded, compared to the same molecule after some time it has passed and the molecule has interacted. You can see, number one, that there are many more interaction sites with the substrate, and the molecule has changed its configuration. It has become longer and more and flat. Going back to table 3.1 and continuing our review of the properties of proteins which affect their interactions with substrates, consider now structure. First and foremost, the stability of the proteins is very important. It is known that proteins which are not very stable, even if they make the first contact and link with the substrate surface, they may not stay there for a long period of time. Another important aspect to keep in mind is the following. Proteins are bioactive chemical compounds. They are therefore depending on various aspects of the environment in which they find themselves. And the biological, physiological environment has its own pH, it has its own temperature, even under normal conditions, never mind under conditions of high fever when the temperature, of course, of the human body increases. And other situations which may affect the stability of the molecules of proteins. As a matter of fact, it is known that depending on the pH and the temperature, proteins may become denatured, which means they lose their bioactivity. And this is something that we have to keep in mind when we are considering protein interactions with biomaterial surfaces. Another aspect which is very important and unique regarding the structures of proteins is the so-called unfolding rate. Once the protein interacts and it is given some time to make more contact points and spread out, the protein also unfolds. This particular aspect is very convincingly shown in the cartoon of figure 3.3. On the top layer you see a protein, and it's usually represented as a convoluted big spaghetti, if you wish, making some contact with the substrate. But as time progresses and many more contacts are made by that protein with molecule with the substrate surface, you can see that particular structure spreading out and unfolding. This particular phenomenon is known as protein conformation. It refers to a specific three-dimensional configuration or configurations that the protein structure can acquire when the proteins adsorb on the surface of a substrate. As I mentioned earlier, major advances in research regarding protein interactions with substrates were achieved using very dilute concentrations of proteins. Keep in mind, however, that even then proteins in water, and remember, body fluids are mostly water, proteins in water are heavier than water. Therefore, even under only gravitational forces, the protein molecules slowly but for sure will settle, and some of them eventually will come in contact with the surface of the substrate material. This particular process is shown on the upper layer of this figure 3.4. As time progresses, many more of those molecules in the supernatant solution will land on the surface of the substrate material, and this situation is illustrated in the middle layer of figure 3.4. Eventually, and at much longer periods of time, many more molecules will arrive, but they will find themselves in a crowded situation because other molecules have already arrived and occupied the surface area of the substrate. As a result of that, we're going to have either squeezing of the proteins and therefore different orientation of the molecule of the proteins on the surface, and sometimes some of those molecules may be squeezed off and find themselves in the supernatant bulk solution. Definitely, the extent of the concentration of the protein in the solution will affect its subsequent interaction of the protein molecules with the substrate, as shown on the upper level of figure 3.8. When the bulk concentration is low, the protein molecules arriving on the surface have plenty of surface area to attach and to spread. However, when the concentration is higher in the bulk solution, the protein molecules which have arrived on the surface of the substrate may find themselves in the need to adopt different configuration. Therefore, we have major conformational changes depending on the concentration of a protein in the bulk solution. So far, we emphasized the adsorption requirement, the interactions of protein molecules with the surface of the substrate. But in the very last slide, I mentioned that depending on how many molecules have arrived and as they are trying to interact with the surface, we may have some not only conformational changes, but some of those molecules being squeezed out and forced to go back into the bulk solution away from the surface. The process of detachment of protein molecules from the surface of a substrate and its return to the bulk phase, supernatant bulk phase, is called desorption. Several studies concerning the desorption of proteins have proven that because desorption requires simultaneous breaking down all the bonds, the points by which a protein is adsorbed on the surface of a material, because of that simultaneous requirement is not really very easy to be achieved, and sometimes it's totally impossible. Therefore, we may say that in most cases, proteins adsorbing on a surface, they will stay attached on that particular surface. The events which take place when protein molecules from multi-component solutions interact with surface of materials has also been investigated. What I mean by multi-component is that we have in one and the same solution more than one type of protein. The events which take place when proteins from a multi-component solution interact with the surfaces of substrates is illustrated in the cartoon of figure 3.9. It is known that certain proteins will be the first ones to arrive on the surface of the substrate. In this cartoon, those proteins are illustrated in green. What kind of proteins will arrive first? If they're big molecular size proteins, or if they are present in higher concentrations compared to the other proteins in the solution. Over time, however, the other proteins will start arriving. Cartoon number 3.9 shows two proteins. So the solution that we have has two proteins. Those now, other arriving molecules, especially if they're in higher concentration, they will compete with the first arrivals on the surface, and eventually they may squeeze the green molecules, the first arrivals, and those are pushed back into the bulk supernatant solution. Figure 3.10 illustrates in a schematic fashion what happens as a function of time when proteins, three types of proteins in a solution, are adsorbing on the surface of the material. And what we see here is that each protein has its own time course of arrival and adsorption on the surface of the material. Protein A is the first one to arrive, and it reaches a maximum at a much earlier time than the other two proteins. As time progresses, protein B is the protein which accumulates more and it's adsorbed in higher amounts, while protein A is not present in such high concentrations. Last but not least, over a long period of time, we notice that protein C, which at the beginning and during the time that protein A is the predominant protein on the surface of the material, protein C is very little, but as time progresses, it is the predominant protein. Eventually, the interactions of any protein, predominant protein, will reach a plateau, what you may call a steady state situation. These particular observations were first noted by a renowned colleague, Professor Vroman, and therefore his name, to honor him for his contributions in science and engineering, his name was given to this particular effect. Therefore, the Vroman effect refers to a hierarchical series of collisions. That means, there is some order and some time sequence. These protein molecules absorb but also exchange when they come from the supernatant, from a multi-component solution to interact with the surface of substrate materials. So it is important to remember that the Vroman effect has the following aspects. It deals with multi-component solutions of proteins and how those molecules come in contact, therefore absorb on the surfaces of materials, but they, as a result of the time course of their interactions, exchange in a hierarchical manner as a result of collisions and other interactions with themselves and with the surface of the material. Professor Vroman studied multi-component solutions. His favorite multi-component solution was, of course, the liquid part of blood plasma. This is the example which is given in chapter 3 of the textbook. Table 3.3 gives us a summary of the various proteins which are dissolved in blood plasma, the liquid part of our blood. And we have different proteins. First and foremost, predominant in terms of its concentration is albumin. And it is a relatively small size protein. Then we have immunoglobulin, the abbreviation I, capital G, small case g means immunoglobulin, and G is the type of globulin, IgG, is a much bigger in terms of molecular weight molecule, but its concentration, although smaller than the concentration of albumin, is still much higher than many other proteins which are dissolved in blood plasma. Going down that list, we see that fibrinogen has a molecular weight of 340,000, and it is present in concentration of 3 milligrams per ml. Complement, one of the complements, complement 3, has molecular weight in the order of 180,000 and a concentration of 1.6 milligrams per ml. There are many more other proteins, including low-density lipoprotein, high-density lipoprotein, immunoglobulin A, antitrypsin, all kinds of other proteins which are dissolved in the human blood plasma. The results of the absorption of some, not all, of the proteins which are dissolved in human blood plasma are shown in table 3.5. The absorption was studied on synthetic surfaces, specifically glass and metal oxide. The results show that albumin, although it's not the biggest in terms of molecular weight protein in blood plasma, but it's the most abundant, that protein was found to absorb first. followed by immunoglobulin G, fibrinogen, factor 12, the surface factor, high molecular weight kininogen. You notice in this particular case we have a protein, fibronectin, which is a predominant protein in the extracellular matrix of tissues, but some small concentrations of it can be found in blood as well. So these particular results provided the evidence that we have different types of phenomena which, as biomedical engineers interested in implantable materials and devices, must keep in mind, because our materials and devices will go inside the human body, which has fluids in which there are many proteins dissolved. They are not solutions of only one protein. They are solutions of different types of proteins. Proteins with different molecular weights, different configuration sizes, and etc., etc. I will continue by considering the protein interactions with substrates from what I call the substrate perspective. Some of the properties of surfaces which affect their interaction with proteins are listed in Table 3.2. Topography is one of those properties. If the surface of the substrate has greater texture, that of course increases the surface area, and therefore it can promote extended, enhanced interaction of proteins with that particular surface. The chemical composition of the surface material is another parameter, because that affects how different proteins will interact or not interact, and to what extent they will interact with that particular surface as a result of the chemical composition aspect. Hydrophobicity and hydrophilicity are two terms which describe the interaction of fluids on the surfaces of substrates. Hydrophilic is a surface which promotes the interaction with water. Specifically, the test done is to put a drop of water on the surface and watch how it interacts. If that particular drop spreads out and it doesn't stay all bundled up, then the surface is classified as hydrophilic. If, however, the drop of water stays all bundled up and it doesn't spread out, then the surface is classified as a hydrophobic surface. In the case of interactions of proteins with hydrophobic substrates, it was found that the hydrophobic surfaces tend to bind more protein. If the surface is heterogeneous, that means that's non-uniform characteristics of the surface, that might result in interaction of proteins differently in different domains of the surface of the substrate. An additional property is related to the electrical characteristics of the surface, therefore potential. The charge on the protein molecules is a parameter which will dictate whether a protein will interact with the substrate, but at the same time the charge or the potential of the surface is the other parameter which will bring about or will prevent that particular interaction. This particular situation is illustrated in figure 3.5, which I discussed a little bit earlier in this lecture. And here we have both the molecules of the proteins, which may have different overall negative or overall positive, but even specific distribution of the charge all across their domain. And in addition, the surface of the biomaterial may have domains of different charges, positive or negative, etc. These two different potentials will dictate the interactions. If we have both positive or both negative protein surface of the material, that interaction is not going to take place. We have to have, again, one negative and one positive potential in order to see that interaction take place. This is the end of lecture 15b with the title Protein Interactions with Substrates.